Hello guys, welcome back to my EduSpace. Hope you all are doing and learning well. So in today's video, we are going to look at another cell organelle that is mitochondria. So without further ado, let's get started. First, let me introduce ourselves like what is mitochondria. Okay, so singular, it is mitochondrion. But if we are talking about more than one mitochondrion, we use mitochondria. So mitochondria is a plural form of mitochondrion. Okay, so this word is taken from Greek word which combines two words, that is mitos. The first word is mitos, which means thread, and another word is chondrion, which means granule. Okay, so inside the cell, the mitochondria looks like granules, thread like granules. Suppose if this is our cell, okay, and at the core the nucleus is present, this is nucleus, and there are some other organelle, ER, endoplasmic reticulum, Golgi apparatus. Okay, so if we see, there are small thread-like granules are present all over the cell. These granules are mitochondria. These granules are mitochondria. Okay. So it is round to oval shaped and having double membrane structure. Okay. And found in eukaryotic organisms. So this cell organelle is found in eukaryotic cells, and the structure is oval shaped. Sometimes it may be round, and it is double membrane structure. So you can see in this picture, these are oval shaped or cylindrical shaped structures. These are mitochondria, which are enveloped by double membrane structure. Okay. So mitochondria generate most of the chemical energy needed to power the cell's biochemical reactions. Why our cells are blessed with these small organelles? Because they help in producing energy which is required by a cell to perform its activity. So the energy that is produced by these mitochondria is stored in a small molecule which is known as adenosine triphosphate molecules, or we can say ATP. Next up, the history about mitochondria. So the Albert von Kolliker were first discovered these mitochondria in 1857. Okay, and Richard Altman in 1890 established them as cell organelles. They called them bioblast or life germs. In 1898, Carl Benda renamed these as mitochondria, or the Carl Benda coined the term mitochondria in 1898. As we all know, we say mitochondria are powerhouse of the cell. Then who coined the term powerhouse of the cell? The Philip Sienkiewicz in 1957 coined the term powerhouse of the cell for mitochondria that we are used. Now, how these mitochondria are originated. The origin of mitochondria. For this, two hypotheses were proposed. The first one is endosymbiotic theory, and the another one is autogenous theory. In these two theories, the most accepted one is endosymbiotic theory. In autogenous theory, it was hypothesized that mitochondria were originated by splitting off a portion of DNA from nucleus of an eukaryotic cell, and this DNA would have been enclosed by membrane. So this autogenous theory says that eukaryotic itself originated these mitochondrial structures. Suppose if this is an ancestral eukaryotic cell, okay, having a nucleus and which enclosed genetic material DNA. So what autogeneous theory says? It says this is splitting of some part of DNA. So there is a splitting of some part of DNA, okay? Some part of DNA gets split off. This portion gets split off and it forms ultimately a mitochondrion structure. So this is nucleus having DNA and there is another mitochondrion having its own DNA. So autogeneous theory says that it is splitting of DNA of an eukaryotic cell. Whereas in endosymbiotic theory, mitochondria were prokaryotic cells that can perform aerobic respiration that were caught inside an eukaryotic cell or eukaryotic cell engulfed such prokaryotic cell. Let me discuss more about the endosymbiotic theory of mitochondrial origin because it is highly accepted nowadays. So, this theory hypothesized that mitochondria were prokaryotic cell. Okay, the mitochondria were initially a prokaryotic cell that performed aerobic respiration that were caught inside an eukaryotic cell or an eukaryotic cell engulfed such prokaryotic cell and they form a symbiotic relationship therein inside the cell. That's why it is an endosymbiotic theory. Endo means inside, okay? Endo means inside and symbiotic relationships means the relationships where both receives benefits. So look at this picture. It will be clear all your doubt regarding the endosymbiotic theory. This is an ancestral eukaryotic cell where the cell organelles are in developing stage. Okay, the cell organelles are about to form. So these are internal membranes. These are the internal membranes inside this ancestral eukaryotic cell. We have internal membrane system, this, which later form an endoplasmic reticulum. Okay, so this cell was thought to engulf an aerobic bacterium. This is an aerobic bacterium. Then the prokaryotic cell formed an endosymbiotic relationship with this ancestral eukaryotic cell. So, as it is thought that an ancestral eukaryotic cell engulfed an aerobic bacteria. So, when it comes inside this cell, it forms an symbiotic relationship with this ancestral eukaryotic cell and finally form as an mitochondrial structure. This theory is also true for the chloroplast, but in the chloroplast case, the bacteria was photosynthetic bacteria. So hope you understand. But like people us, we need evidence to prove something. So just like that, many critics criticized this endosymbiotic theory. So there are some evidences that proves this theory correct. So what are these evidences? We will see in next slide. Evidences of endosymbiotic theory. There are three pieces of evidences of the endosymbiotic theory. The first one is self-replicating mitochondria. So the mitochondria are self-replicating just like a prokaryotic cell. They both reproduce by fission. Okay. So as we know that prokaryotic cell is going to reproduce with the help of binary fission, and this is also true for the mitochondria. The mitochondria also reproduce with the help of binary fission. Now the DNA. The DNA of mitochondria shares similarities with the prokaryotic genetic material. Okay. And last, the mitochondria is double membrane structure as we see in the prokaryotic cell. Coming up next is structure. What is the structure of a mitochondria? So you can see this diagram. This is a mitochondria. Okay. The mitochondria may have a number of different shapes. It is not certainty that the mitochondria should be of oval shape or it is cylindrical shape or round shape. The shape varies. Okay, a mitochondrion have double membrane organization and consist of 5 distinct parts. So the structure of mitochondria consists of 5 distinct parts. These parts are the outer mitochondrial membrane, the inner membrane, and the intermembrane space and inner mitochondrial membrane, cristae and matrix. Okay, so look at this diagram. You can see the outer envelope of mitochondria is double membrane structure. The outer one is known as outer mitochondrial membrane. This is our outer mitochondrial membrane, and inside which this space, this is our intermembrane space. Okay, the space that is present between the inner mitochondrial membrane and the outer mitochondrial membrane is known as intermembrane space. And the third one is inner mitochondrial membrane. These infolding like structures, okay, the blue which is shown in blue color, this particular is intermitochondrial membrane, and this is very important in function wise in mitochondria. Next one is cristae. You can see these folding like structure, these infoldings of the inner membrane are known as cristae. Basically, they expand the surface area of the inner mitochondrial membrane. And the next and the last one is the matrix. The thing that is present inside the inner mitochondrial membrane is known as matrix, or we can say this is the space within the inner membrane which is a fluid like. Okay, outer membrane, or we can say outer mitochondrial membrane. The outer covering of the mitochondria is known as outer mitochondrial membrane. This is known as outer mitochondrial membrane. Okay. It surrounds the complete structure of a mitochondria. Basically, it is 60 to 75 angstrom thick. This particular membrane is thick about 60 to 75 angstrom. It is made up of protein phospholipid bilayer. Okay, the main constituent of this layer is protein and phospholipid bilayer, just similar to the cell membrane. It contains many integral membrane proteins called porins. So the surface of the outer membrane consists of so many proteins that are known as porins. What are the function of porins? We already discussed it, that these porins allow transport of ions and molecules, small molecules and ion without any hindrances. Okay, they does not require any special proteins to transfer. Basically, these porins allow transport of small molecules or ions easily inside or outside the mitochondria. Okay, a major trafficking protein is the beta barrel. Now the porins are obviously it is made up of transporting proteins. Now this transporting protein is basically beta barrel pore forming. Why this is called pore forming? Because it forms pore or holes over the surface of the outer membrane. So this particular beta barrel pore forming protein is voltage dependent anion channel. This is VDAC. This particular porine is known as VDAC, which means voltage dependent anion channels. The VDAC involves in transporting nucleotides, ions and metabolites between the cytosol and the intermembrane space. So these porins helps in transporting nucleotides, ions and metabolites across these outer membrane to this intermembrane space. So if these porins are allow all the ions and molecules to pass across this, so the concentration of the intermembrane space, here the concentration of ions and molecules in this intermembrane space is almost similar to that of cytosol. Okay, to that of cytosol, because these porins allow of transporting all the ions, small ions, metabolites across this outer membrane space. So we had just discussed about the outer membrane of mitochondria having multiple of porins or VDAC. Okay, and these VDAC allows small molecules and ion to pass from the cytosol to the intermembrane space. Okay, it allows molecules or ions to pass from the cytosol to the intermembrane space. But small molecules and ions, metabolites can easily pass through porins, but it's found difficult to pass large proteins across outer membrane. So, large proteins need some extra effort to pass across outer mitochondrial membrane. So, if a large protein has to be transported across this outer membrane, so what it need to do? We know that all the polypeptide chain having an amino terminus, okay, and a carboxy terminus. Okay, so the signaling sequence at N-terminus of large protein must bind to the large multi-subunit protein called translocase. So suppose this is a very large polypeptide chain, and at the N-terminus, at the amino terminus, and there is some sequence which is known as signaling sequence. Suppose this particular sequence is the signaling sequence that is present at the N-terminal end of a polypeptide chain. So this particular end has to be bind to a multi-subunit protein that is present on the outer mitochondrial membrane, and this is known as translocase. Okay, this is translocase. This is translocase protein. So this large polypeptide having a signal sequence at its N-terminus now get bind to the translocase. Okay, now this polypeptide is going to bind here. This is carboxyline, and here this is N-terminus. This particular signaling sequence that bound to the translocase. Now this translocase at the outer membrane actively moves this large protein cross the outer membrane to the intermembrane space. Now it can easily, this translocase protein can easily pass this large protein molecules to inside the intermembrane space. Okay. I just told you that it actively moves. So what does it mean? It actively moves means it requires energy. Because of this, the protein is not same as that in the cytosol. Okay, so the protein concentration that is present in the cytosol is not equivalent to the protein that is the protein concentration that is present in the intermembrane space. Okay. Now, the most important topic is MAMs or mitochondria associated membrane, abbreviated as MAMs or MAMs. This is outer membrane of mitochondria get associated with endoplasmic reticulum membrane, and these structures are known as MAM. Okay, so there are two organelles, the endoplasmic reticulum and the mitochondria. Okay, they are connected together by lots of protein. You can see these are the proteins. Okay, so they are connected together with lots of protein at the junction of these two organelle membranes. The outer mitochondrial membrane can associate with the endoplasmic reticulum membrane in a structure called MAM. So there are lots of structures that are present inside the cell where the endoplasmic reticulum is associated with the outer membrane of a mitochondria, and they are connected together with many proteins. Okay, so MAPs are the regions of the endoplasmic reticulum that mediate communication between endoplasmic reticulum and mitochondria. Okay, so in this picture you see this is the endoplasmic reticulum and this is mitochondria. The junction of these two cell organelle are studded with lots of protein molecules, okay, that help in establishing communication between these two cell organelle. Basically, it is important in the mitochondria calcium signaling or calcium transport. So basically, the main function of these two, the junction of these two organelle is the calcium ion regulation transport, or we can say in calcium signaling, and also it helps in transfer of lipids between the endoplasmic reticulum and mitochondria, and also last but not the least, it is responsible for the autophagy, or we can say the apoptosis process. Now, as I just told you about what is the outer mitochondrial membrane, the outer membrane of the mitochondria is known as outer mitochondrial membrane. Okay, this is outer membrane of mitochondria. Now, inside this membrane, you can see there is a space, this one, this space. Okay, the space between the outer membrane and the inner membrane. This one is inner membrane. Okay, so the space that is present between the outer membrane and inner membrane is known as our intermembrane space. Okay, so this particular space that is present in between the outer membrane and inner membrane is known as intermembrane space. It is also known as perimitochondrial space. This space lies between inner membrane and outer membrane. The concentration of the small molecules like ions and sugars in the intermembrane space is same as that of the cytosol, because outer membrane is freely permeable to small molecules. As I just told you in the section of outer membrane that the outer membrane contains lots of porins, and these porins are freely permeable to transport small molecules or ions from the cytosol, okay, from the cytosol to inside the intermembrane space. So the concentration of the molecules and sugars ions are almost similar that is found in the cytosol, but the protein concentration at the intermembrane space is varies and is not similar as in the case of cytosol, because these porins are not freely permeable to all the proteins that can pass through the outer membrane of mitochondria. So the large protein required some specific signaling sequence to get transported across the outer membrane. So the protein composition differs from the cytosol. I just told you that the larger proteins need some signaling sequence which is present at the N-terminal end of a polypeptide chain or of large molecule of protein. So with this signaling sequence, it can binds to the translocase that is present on the outer membrane, and then only the large protein can pass from the cytosol to the intermembrane space. So it is not freely permeable to all the proteins, thereby the protein concentration is differs from the cytosol. Okay. The next one is inner membrane. So after the outer membrane and the intermembrane space. Okay, this is intermembrane space, this is outer membrane. Here the comes inner membrane. Okay, this one is the inner membrane, the lining of the inner membrane. So it is rich in protein content. About 151 different polypeptides are found. It contains around 1/5 of the total protein in a mitochondrion. So this inner membrane is rich in the protein content. There are about 151 of different proteins are present in the inner membrane and which are having different functions. Okay, it is rich in special phospholipid that is known as cardiolipin. As I just told you, the outer membrane is the concentration or the constituent of the protein and phospholipid are almost similar of the outer membrane with that of the cell membrane. But in the case of inner membrane, the concentration, or we can say the protein and phospholipid concentration, is not similar to that of the cell membrane. In this, a special type of phospholipid, this is known as cardiolipin, is present in the inner membrane. We know that phospholipid is of this type, having two fatty acid chains. But in case of cardiolipin, the structure is somewhat difficult. The structure is having, this is our hydrophilic head, and it having 4 fatty acid chains. Consist of 4 fatty acid chains. Okay, the cardiolipin having 4 fatty acid chains that makes inner membrane impermeable. Because of this, they are having long 4 fatty acid chains. It makes membrane, the inner membrane, impermeable to anything, that is maybe it ions, molecules. So it does not contain porins, which so thereby impermeable to all ions and molecules. For transporting the ions and molecules, it needs special membrane transporters which help them to transport to the intermembrane space to the matrix, okay, or from the matrix to the intermembrane space. So the molecules or ions cannot easily pass across the inner membrane from the matrix to intermembrane space or from the intermembrane space to the matrix. As I just told you that inner membrane or the inner mitochondrial membrane contains about 151 different types of proteins. So they have different specific functions. The inner mitochondrial membrane contains proteins having specific function. So we can divide these proteins according to their function in three parts. Okay. Some proteins perform electron transport chain redox reactions. Okay. Some proteins are responsible for performing the electron transport chain. Some proteins are known as ATP synthase that forms ATP in the matrix. So some proteins that are present on the inner membranes are known as ATP synthase. So these are ATP synthase. These are ATP synthase which help in the formation of ATP. They form ATPs. Okay, the ATP synthase forms or generate the ATP. Now the subprotein fraction is known as transport proteins, as its name suggested that these proteins are responsible for transporting metabolites into and out of the matrix. So some proteins known as that are present in the inner membrane are known as what? They are known as transport proteins. Okay, these are transport proteins. These are transport proteins. What they do? They basically helps in transporting the metabolites into and out of this matrix. Into or out of this matrix. Okay. So the next one is Cristae. Singular, it is known as Crista. And if we talk about more than one Crista, we say it is Cristae. Okay. So the inner membrane is having numerous infolds, which is known as Cristae. You can see this one is the outer membrane and this one is the inner membrane. So the inner membranes are having lots of infolds inside the matrix. Okay. You can see these are the infolds. Sometimes these infolds are tubule-like. Okay, that we can see over here in this picture. These are tubules-like infolds. Okay, basically it expands the surface area of inner membrane, as we just discussed in the previous slide that the inner membrane contains 151 different types of proteins that have different and specific functions. So for enhancing these functions, the surface area has to be increased. So the inner membrane is having lots of infolds, which is known as cristae. Thereby, it also enhancing its ability to produce ATP, okay, adenosine triphosphate molecules. With the increase in the surface area, there are lots of ATP synthase that can present in the membrane. So thereby, with lots of ATP synthase, the ATP production is also increases. The ratio of the Cristae per mitochondrion is variable as it depends upon the demand for ATP in a cell. So it is not certain amount of number of Cristae are present in the inner membrane. The number or the ratio of the Cristae per mitochondrion is varies, and basically it depends upon the demand for ATP in a cell. If a cell is in requirement of more energy, then the Cristae number get increases. Okay. So the mitochondria from cells that have a greater demand for ATP, such as muscle cells. So the muscle cell contains more Cristae as compared to other cell. The mitochondria within the same cell can have different Cristae density, with the ones that are required to produce more ATP having much more Cristae. So it is not in the case of different type of cells. In a single cell, if we see that there are two mitochondria are present, two or many more we can say, but here we are taking examples of two mitochondria. Okay, so if this mitochondria is responsible for forming more ATP synthesis, then the number of Cristae inside this is much higher as compared to this mitochondria. So the mitochondria within the same cell can have different Cristae density. Okay, some that are required to produce more ATP are having much more Cristae. The Cristae are studded with small round bodies known as F1 particles or oxisomes. Okay, so these Cristae, they are studded with FOF1 particles, or we can say oxisomes, that is responsible for oxidative phosphorylation. Okay, so the last part of the mitochondrial structure is matrix. This is the space which is enclosed by inner membrane is known as matrix and contains about 2/3 of the total proteins in a mitochondrion. So if this is our inner membrane, okay, so this particular space, this space is known as matrix. This particular space is known as matrix and it contains lots of proteins, about 2/3 of the total proteins in a mitochondrion. Basically, it facilitates production of ATP with the help of ATP synthase that is present in the inner membrane. So, we know that in the Cristae, we have in the Cristae and in the inner mitochondrial membrane, we have ATP synthase which help in the production of ATP. As we know that these mitochondria are self-replicating. So how they replicate themselves? Because they have their own DNA. So you can see in this diagram, these thread-like structures, these are mitochondrial DNA. Okay, these are mitochondrial DNA. And as they are self-replicating, if they have their own DNA, then obviously they have their own ribosome. So these small structures are known as ribosomes. So we can say that matrix contains a highly concentrated mixture of hundreds of enzymes and several copies of mitochondrial DNA. So these are the mitochondrial DNA, and there are lots of enzymes that are present inside the matrix. There are mitochondrial ribosomes are also present and the tRNA. Okay, so the matrix contain what? They contain hundreds of enzymes, several copies of mitochondrial DNA, several copies of mitochondrial ribosomes, and the transfer RNA. Now, what are the functions of this matrix? Basically, in the matrix, there is an oxidation of pyruvate and fatty acids takes place, and the citric acid cycle, or we can say the Krebs cycles, also takes place in the matrix. So, as we just discussed how strong and powerful a mitochondria is. So, there are lots of functions, but here I am presenting some functions of mitochondria. So, it uses oxygen which is available within the cell. It converts chemical energy from food to energy in a form of ATP, which is usable to the host cell. So what the mitochondria does, basically it converts the chemical energy from food to energy that is usable by the host cell. Basically, it is in the form of ATP, that is adenosine triphosphate. This process is known as oxidative phosphorylation. The mitochondria also helps in cellular metabolism. It helps in citric acid cycle or Krebs cycle. It helps in producing heat, and also it helps in regulation of calcium ion concentration that we had just discussed in MAMs, okay, and it also helps in the production of certain steroids. Thanks for watching.